So once you kind of have a good idea of, a target and a specific, pharmacological mechanism that you're working with, then that's when you can start kind of going through the drug approval process. And so that's when we kind of get into the preclinical phase of drug discovery, where people are doing more animal model testing. So typically with animal models, you are really looking at studying the medication in two different animals, and the kind of purpose of doing the animal models is, of course, we don't want to expose humans to something toxic or potentially dangerous prior. And it allows us to ensure that there's some biological activity in the animal model. It can also start running some of the additional screening methods. So we can really look at stability testing, animal safety, kind of start developing lethal dose ranges, the toxic ranges. Is it a wide enough effect? Because every potential medication, there is a lethal dose and there is a effective dose, and that goes for probably any type of biological compound, even things like water. We know that water has actually a lethal ingestion if you drink too much, and it is required though for sustaining natural life. So some of the other things that we do in the preclinical animal studies is focusing in on some of the pharmacology, so the mechanism, making sure that it is the actual mechanism of action. We do toxicity studies. We do two different animal models, so generally one rodent, one non-rodent. There has been in the past where drugs have been studied in a rodent model, and certain adverse effects weren't discovered because of that. So it's imperative to have two biological systems. And then we can kind of get a good idea of the pharmacokinetics, or at least some baseline data on the pharmacokinetics. So that's really what the body does to the drug. So metabolism, excretion, the volume of distribution, how is it absorbed. And animal testing, it really provides the initial data for this. And if you have good animal data testing, if things are positive that you don't really have toxicity associated with, you can go to the next forms of testing. And so that's when you would start your human clinical study.